Hello students, I am Mrs. Zuili Pankaj Dhokne, working as Assistant Professor in KSKW Art, Science and Commerce College, Sidco, Nasik. Today we are going to see the topic Streptococcus. Streptococcus is gram-positive cocci that grows in pairs or in chains. It is the normal flora of human and animals. It inhabits in the various sites of the body. Notably, it is present in the upper respiratory tract and live harmlessly as commensals in the body. The most important organism of the genus Streptococci, that is Streptococcus pyogenes. This Streptococcus pyogen, it is mainly responsible for causing pyogenic infection with a characteristic tendency to spread. The lesions with this Streptococcus pyogenes, they are spreading type of nature, as opposed to staphylococcal lesions. Staphylococcal lesions, we know that they are localized lesion. So if we compare the lesions from Streptococcus pyogenes and staphylococci, there is a difference that in case of Streptococcus pyogenes, the lesions are spreading, and in case of staphylococci, the lesions are localized. This Streptococcus pyogen, it is also responsible for the non-suppurative lesions, that is acute traumatic fever and glomerulonephritis. These two infections caused by Streptococcus. In this infection or in this type of disease, in rheumatic fever and glomerulonephritis, there is no pus forming lesion on the body, but the symptoms are very severe. In the image given in the image here, it is showing the streptococci present in chain. If we see the classification of streptococci, so streptococci are first divided into obligate anaerobes and facultative anaerobes. So obligate anaerobes are designated as peptostreptococci. So there are three different schemes which are used for classification of this organism. The basic classification of Streptococcus genus, it is done on the hemolytic patterns produced by bacteria on the blood agar. So hemolytic pattern, that is the bacteria or the streptococci which is showing the complete zone of hemolysis, that is known as the beta hemolysis, and the one which is not responsible for hemolysis, that is no hemolysis or gamma hemolysis. So based on this classification or based on this hemolysis, the streptococci, they are classified as hemolytic streptococci and non-hemolytic streptococci. Serological properties of this bacteria, they are studied and they are classified by Lancefield. The classification of Lancefield, that is Lancefield grouping A to H, K to M and O to V except I and J. Also the classification, it is done based on the biochemical properties. If we see the classification of streptococci, this is the Lancefield classification. So first classification, it is done based on oxygen requirement. Aerobes and facultative anaerobes, these are the classes or these are the class of streptococci which is aerobic and facultatively anaerobic, and the other is obligate anaerobes, they are known as peptostreptococci. If we see or if we further classify the aerobes and facultative anaerobes, so based on hemolysis, as we saw in the previous slide, some streptococci, they are showing the alpha hemolysis. It means that complete hemolysis doesn't occur on the plate, only the partial degradation or partial lysis of RBCs, it occurs on the plate, and the color of the blood agar, it becomes greenish, and that type of hemolysis is known as alpha hemolysis. Also this alpha hemolytic bacteria, they are known as the viridians group. Again, second classification, that is based on the complete hemolysis, that is beta hemolysis. beta hemolysis. These are the special type of streptococci, that is the hemolytic streptococci, and they are further classified based on the serological grouping, that is on polysaccharide C antigen present in the cell wall. Based on this polysaccharide C antigen, there are 20 Lancefield groups from A to V except I and J. A group, it mainly consists the pathogen, that is Streptococcus pyogenes. Streptococcus pyogen, which is responsible for the pyogenic infection, also the rheumatic fever and glomerulonephritis. Again, this group A streptococci, that is Streptococcus pyogen, it is further classified based on the M protein, and this M protein, it is based on this M protein, it is classified into 18 serotypes from 1 to 80, and that classification is known as Griffith's typing. Obligate anaerobes, that is peptostreptococci, as they are not involved in pathogenicity, so they are not further classified here. In this image, we can see what is alpha hemolysis, what is beta hemolysis, and what is gamma hemolysis. So A, which is showing, in A, you can see the complete or clear zone around the colonies, that is the RBCs, they are completely lysed, and that is the beta hemolysis. If you see B, the area around B where RBC, they are covered by the greenish discoloration zone, that is the beta hemolysis, and in case of C, there is no hemolysis, the colonies are as it is, and there is absence of any zone around the colony, that is the no hemolysis, that is gamma hemolysis. If we see, now we will concentrate on the main pathogen from this Streptococcus genus, that is Streptococcus pyogenes. So, if we see morphology of Streptococcus pyogen, it is gram positive. Then, if we see the shape, it is cocci, and the size from 0.5 to 1 micrometer in diameter. It grows in short or moderately long chain. If we see the image which is mentioned in the second slide, then we can see that how they are present in the chains. The chain length depend on the strain and culture medium, in which medium they are growing, whether they are growing in the liquid medium or on the solid medium. Depending on that, the length of the chain, it is dependent. Chain formation is due to the cocci dividing in one plane, only the daughter cells failing to separate completely. The chains longer in liquid media than in solid media. Non-motile, non-sporing, and few strains are capsulated. So, the streptococci, they are non-motile, they also do not form spore, and few strains of this streptococci, they are capsulated. Some strains of the Streptococcus pyogen and some group C strains produced a capsule of hyaluronic acid. So, in bacteria, the capsule, it is made up of different material. Sometimes it may be polysaccharidal capsule, sometimes it may be protein capsule. So here in case of Streptococcus pyogen, the capsule is made up from hyaluronic acid. Cultural characteristics of Streptococcus pyogen. The first thing, it is aerobe and facultative anaerobe. Optimum temperature for growth of this Streptococcus pyogen, it is 37 degrees centigrade. It can grow in the range from 22 to 42 degrees centigrade. Optimum pH for growth, it is 7.4 to 7.6. And exacting the nutrient requirements of this organism made this organism special, that it requires in the media with fermentable carbohydrates or the media which is enriched with blood. So this blood agar, it is used for growth and isolation of Streptococcus pyogen. Blood agar. So if you see the colonies on blood agar of this Streptococcus pyogen, the image, it is there. In the image we can see the diameter of the colony, it is from 0.5 to 1 millimeter. The shape of the colony is circular. They are semi-transparent, low convex discs, and they are surrounded by a wide zone of hemolysis. So several times greater than the diameter of colony After incubation for 24 hours, so you can see in the image, see the zone of hemolysis, it is 2, means the size of the colony, it is very small, and as compared to the size of colony, the zone of hemolysis, it is large. So beta hemolysis, it is the primary marker for isolation of Streptococcus pyogenes, because other streptococci, the hemolytic streptococci, in that group A streptococci, which is Streptococcus pyogenes, it is showing the complete zone of hemolysis or beta hemolysis. If we incubate the plates at 10% CO2, then definitely the hemolysis zone or zone of hemolysis, that will be more, that is the hemolysis process, it will be promoted at 10% CO2 incubation. Now, the cultural characteristics of this bacteria, that they are forming the matte or finely granular colonies, and these colonies, they are containing M antigen, which is a virulent strain, while avirulent strains from glossy colonies. So virulent strain, they are matte, and the avirulent strain, they are glossy. Then mucoid colonies may occur when there is this strain which is highly capsulated, or the strain which is producing high capsule in high concentration. That colonies, they are mucoid colonies. So the media used for the growth of the hemolytic streptococci, the selective media, that is the crystal violet blood agar. Then the PNF medium, in the PNF medium there is blood agar added with polymyxin B, neomycin and fusidic acid. Also the transport medium for the streptococci, that is Pike's medium. So the clinical samples which are having the group A streptococci, and if the distance of the laboratory, it is long, or if it takes long time to get the for the isolation process, at that time the sample or the specimen which you have collected from the patient, it is put into the Pike's medium, and then it is sent to the laboratory. So this Pike's medium, it is prepared by adding crystal violet and sodium azide to blood agar. Liquid media such as glucose or serum broth, growth occurs as a granular turbidity with a powdery deposit in this media, which is having the glucose or serum. In this media, the growth of streptococci, that is granular turbidity with a powdery deposit, and in case of streptococci, no pellicle is formed. If you see biochemical reactions, Streptococci is catalase negative. Also, if you put the colonies into 10% bile, it will not get soluble like pneumococci. Ferment several sugar, and by fermentation process it will produce acid but no gas. If you see resistance of this streptococci, it is highly delicate organism. It is killed by heating at 54 degrees centigrade for 30 minutes. Also, it survives in the dust for several weeks if it is protected from sunlight, as it is delicate. In that for that delicate organism, if you want to survive, or if that organism want to survive, it has to be protected from sunlight. So in the laboratory culture, it is stored at 3 to 5 degrees centigrade in blood broth or cooked meat medium, or it can be freeze dried. This organism, it is sensitive to most antiseptic and is rapidly inactivated by crystal violet, nalidixic acid and colistin sulfate. When these antibiotics or the dye, they are added to blood agar, they are providing the good selective medium. So if we want to grow the bacteria streptococci, in that or for that one medium we saw, but if we want to make the medium selective, then we can use nalidixic acid, crystal violet and colistin sulfate in the blood agar, and that will be provide good medium. But the bacteria, it is sensitive to most antiseptics and is rapidly inactivated. Cross reaction of human tissue with Streptococcus pyogenes component. So as we know, the cross reactivity is one of the property of the body in which the body, it produces antibodies against the antigen, but the nature of the antigen of the body. It is similar to the pathogen antigen, so that antibodies which are produced by the body, which are acting on the antigen or pathogen, at the same time they are affecting our body. So that is known as cross reaction. So Streptococcus pyogen having its cell components which are showing the cross reaction with the body tissues. So the first is the capsular hyaluronic acid present in the streptococcal cell wall, or which is present outside the cell wall. It is having the similarity with the synovial fluid. So the antibodies which are synthesized against the capsular hyaluronic acid will act on the synovial fluid. Also, the cell wall protein having the similarity with the myocardium. So the antibodies which are synthesized for the cell wall protein, they will act on myocardium. Cell wall carbohydrates having the similarity with the cardiac walls, cytoplasmic membrane which is similar to the vascular intima, that is the internal layer of the blood vessels, and peptidoglycan, it is having the similarity with the skin antigen. So here the cross reactivity between the bacterial antigen and between the human cells or human tissues, that is mentioned. So all these cases, when the antibodies they are synthesized to all these antigen they will simultaneously act on the our tissue like synovial fluid, myocardium, which is the myocardium, it is the middle layer of heart. Then cardiac walls, that is these walls which are mainly regulating the blood inflow and outflow. Also the vascular intima and skin antigen, all will get affected if the person get infection from the Streptococcus pyogen. If you see the antigenic structure of this bacteria, so here in this image we can see the antigenic structure. Inner layer of peptidoglycan, that is the peptidoglycan which is responsible for cell wall rigidity, it is present in the innermost layer. The biological property which is shown by the cell wall peptidoglycan, that is it is mainly responsible for pyrogenic and thrombolytic activity. Middle layer of group specific carbohydrate, serological classification of beta hemolytic streptococci is based on their cell wall polysaccharide antigen. So this cell wall antigen, it is the integral part of cell wall and it has to be extracted for grouping by a precipitation test with group antisera. A variety of techniques have been used for the extraction of the group antigen. So Lancefield acid extraction method, which is used for extraction of antigen. In this method, the streptococci they are grown in the Todd habit broth and extracted with hydrochloric acid. Also, Fuller's method, then Maxted's method, these methods they are used for extraction of antigen. Also the antigen they can be used or they can be extracted by autoclaving. Outer layer of protein and lipoteichoic acid containing protein antigens, that is M, T and R. So the outer layer of Streptococcus pyogen, it is mainly having the three antigens, M antigen, T antigen and R antigen. M protein, it is associated with the virulent streptococci and virulence factor by inhibiting phagocytosis. It inhibits phagocytosis. Antigenic and specific anti-M antibody develop after infection, which enhances the phagocytosis of the coccus and it is protective. M protein is resistant to heat and acid but susceptible to trypsin. It can be extracted by the Lancefield and extraction method. T protein, that is the trypsin resistant protein, is heat and acid labile but resistant to trypsin, present in many serotype of S pyogen. It may be specific, but many different M type possesses the same antigen. T typing is performed by a slide agglutination test that uses trypsin treated whole streptococci. T protein has no relation to virulence. R protein, some R strains of Streptococcus pyogens and some of group B, C and G contain third antigen R protein. So R protein has no relation to virulence. M associated protein, that is MAP, a non-type specific protein associated with the M protein has been identified and this is known as M associated protein. The capsule composed of hyaluronic acid, which is non-immunogenic, antiphagocytic effect like other bacterial capsule. This is the structure of capsule. In this capsule structure, we can see the cell membrane, the capsule hyaluronic acid, peptidoglycan layer, group carbohydrate antigen C, and lipoteichoic acid layer. The fimbriae are also mentioned here. The capsule composed of hyaluronic acid, not immunogenic, antiphagocytic effect like other bacterial capsules. So we will stop here. Thank you.